So that area of biotechnology called genetic engineering is where we headed into in our story. And yesterday we covered the 3 key tools that a biotechnician or genetic engineering scientist works with. So I just want to, by diagram, recap our story of yesterday. So we start off, scientists know the cells that the gene that they're trying to manipulate, put somewhere, do something with. They find those cells in whatever tissue it is. For example, that could be human cheek cells if we're looking for the insulin gene. It could be the cells from a fish from Antarctica if we're looking for a gene that gives a bit of frost resistance to those fish. So there's a whole lot of ones. It could be daffodil or capsicum cells, and in there is the vitamin A gene. So there's a whole lot of different uses for this idea of finding genes. We're going to look at 2 examples. We're going to look at the example of insulin, being able to produce insulin. So we're trying to find the insulin gene, which means we would use human tissues. Then another example that we're going to be talking about is the vitamin A gene. And for this, the scientists wouldn't use human cells because we don't have the vitamin A gene in our cells. What they actually do is use a combination of cells, but daffodil cells in particular, because those cells have this vitamin A gene in it. So we have the cells that have the gene that the scientists are wanting to isolate. So the first thing that happens is the DNA is extracted and placed into a test tube. And in that DNA is every gene of that particular species. So if it's the insulin gene we want, in that DNA is every gene for every human feature, including the insulin gene. So we now put that DNA into our test tube. And in order for us to actually get our specific gene, the insulin gene or the vitamin A gene, we have to add those very special enzymes, the first of our key enzyme groups, and they are the restriction enzymes. Which I usually abbreviate to RE. That is not an official abbreviation. RE is just Juliet's abbreviation. So we work out the specific one that we need because it has a particular recognition site, which is going to get our DNA molecule. And because we know what bases are at the end of our genes, this restriction enzyme is going to cut the combination of the ends, which will then us allow to have our gene of interest, or else often called the target gene. So that would be our insulin gene, or that would be our vitamin A gene. So that was step 1 in our tools of how we start. So now what we've got is all this cut up DNA, which is often referred to as the digested or sample DNA. It's the one we're now going to need to separate out all the fragments so that we can find ours. So now we go into step 2. And step 2 is to put that cut up DNA into the gel for gel electrophoresis. And remember the gel story, you need to have an idea of the setup of the gel and how the gel works to separate the fragments. So here's our piece of gel. This is looking flat down onto the gel. And at one end we have wells cut. The number of wells depends on how many samples you want to run, but I'll just put 3 there just for the sake of it. So there are our wells. Into our wells, we're going to put our sample DNA. This is the digested one, so that goes in there. And in another well is our standard or marker DNA. And remember, this is fragments of known sizes that we've obtained, and we're using that and we're going to run it at the same time so we have a comparison marker. So in order to run the gel, we have to connect the electrodes. The negative electrode gets connected at the well end and the positive electrode at the other end. Because we know DNA is negative charged. And therefore, when we turn the gel on, the DNA is going to be attracted to the positive electrode. We also know the size of the fragments is going to play a bearing in how fast they run through the gel. So we know the smaller ones will run faster and further in the time given compared to the larger ones. So when we turn it on, the DNA, our standard, will separate into the pieces based on size. So will our sample, depending on where the DNA was cut and how many fragments we've got. Remembering, the one that's gone the furthest is the smallest in base pair numbers, and therefore it's going to move the fastest and therefore the furthest in the whole process, and hence the one up near the well is going to be the larger. The other little bit of information to remind you is that we do have a buffer solution that is made of water and salt, and that's there to make the whole gel run even better. So we run the gel based on those two DNA properties. But remember, there are things that can also impact, like how much current we put on and how dense or thick the gel is, etc. So in doing this and using our sample, and yes, I forgot to mention there's always a little dye color molecule. So we'll just put the little dye front molecule down there to tell us when to turn it off so that it all doesn't run. Then using our stain and being able to tell which fragment is ours, and I'm just going to pretend that is our insulin gene, if this was human DNA, or that is our vitamin A gene, if this was daffodil DNA. So this is the way we separate, and gel electrophoresis is for separation. Retsriction enzymes are for cutting the gene out of the DNA. Gel electrophoresis is just to separate the fragments. And then finally, because we don't tend to have enough DNA, we only have maybe a couple of hundred pieces of our gene, what we need to do is make a lot more copies. So this is where the third tool comes into play. I can't use that color, I'll use this color, and that is the PCR process. And so what we're going to do is have in the PCR our genes that we've cut out. We're going to have the nucleotides, AGTC. We're going to have those special little primers, those little bits of our DNA that will help our enzyme work. And then we have the second of our key enzymes, DNA polymerase. And remember, it's that special one that can withstand really hot temperatures, TAC, DNA polymerase. All that goes into the machine. And then we have 3 steps. Step 1, the machine heats itself up to about 95 degrees. And what we're going to have is the DNA will be denatured. And what that means, we're going to split into 2 separate strands. So the DNA ladder is now separated into 2 separate separate strands for our particular sample that we've put in there. Then the machine cools itself down to about 50 degrees. This is called the annealing stage. Because what's happening here, this is where those primers, those little pieces of DNA, are going to attach to the 3' ends of each strand. So we've got our starting blocks, if you like. And then the third step, it's going to warm itself up again to about 72 degrees. And this is where our magical enzyme, the TAC DNA polymerase, will do its job, where it's going to attach complementary nucleotides to form the new sides, and therefore we're going to have 2 DNA molecules from each one DNA molecule that was put in. And then, so that we can get lots more, it will keep repeating. So if we put in one sample, after one cycle, there'll be 2 lots of it. After another cycle, there'll be 4. After another cycle, there'll be 8, and so on. We have this doubling effect. So after several cycles, we're going to have millions of exact copies of the DNA that we put in. And that, in our case, is going to be the insulin gene or the vitamin A gene. As we'll see, there are other uses for this. And if we're doing something like forensic science, we're not just putting a gene in, we're putting the whole genome DNA. So we will have millions of copies of that DNA. So these are the 3 tools that is used, and we're going to need to know the information sort of at the standard level that I have in this diagram here. And I always find it nice when there are processes like this. We've got 3 separate processes. Sometimes to draw diagrams is a really nice way to learn it. And as I draw the diagram, even as a teacher or a student, I always talk the process to myself. So I'm learning as I'm drawing. And I draw the diagram in sequence of what's happening. Did you notice for the restriction enzymes, I drew the test tube, then I put the DNA in there, then I put the restriction enzyme arrow, then I did the little cuts, because that's the order it happens in. Same with the gel electrophoresis. I drew that picture in the order of the sequence of events to set it up, and same with the PCR. So my advice, where you can, if you like to look at visual pictures rather than lots of words, do these sort of diagrams. It just takes an A4 sheet of paper, and you can have it stuck on your wall if you're allowed to, and therefore it's up there, and you've got this visual memory that you'll be able to take on board as you move through. So that's the story of the 3 tools. So what we've now got at the end of this is a test tube full of millions of copies of our particular target gene. Millions of copies of the insulin gene ready to do something with. Because having a test tube of genes does nothing. The genes don't do anything. They just sit there. Genes only are doing any use if they're in a cell to be transcribed and translated. And so what we're going to do in a minute is go into, well, what do we now do with those genes? How do we get them into cells? Which cells do we put them in? And why do we do it in the first place? So hopefully that's just a little summary of what we went through yesterday. Okay, so from Stephen, are the nucleotides present in the machine, or does the... No. We put those nucleotides in the machine at the start. You can actually purchase solutions of adenine nucleotides guanine nucleotides, thymine nucleotides, cytosine nucleotides. So they're in 4 separate bottles, and the scientist just pipettes some of each nucleotide into the little test tube that goes into the machine. What the polymerase does is it attaches them. It attaches them, for example, if one side of the ladder had an adenine, that's our target gene, the DNA polymerase will put a thymine there. If one side of the ladder had a cytosine, the DNA polymerase would join a guanine. So the DNA polymerase is just finding, if you like, the correct nucleotide and joining it to our separated sides. And that way we can get the copy, the complementary copy. Can the DNA polymerase run out? Well, as we're going to learn, enzymes keep doing their job. They keep repeating their job. Enzymes don't actually do their job and then break down. So they're able to keep doing their job. Of course, there's going to be a limit because enzymes' shape can change a bit, their proteins, and if they change their shape, they're not going to work as well. But in the time sequence we're talking about over several hours of setting up the machine, that Taq DNA polymerase that was put in at the start is still working just as efficiently after several hours, doing its job at joining those nucleotides. And the joining of it, as we're all jumping ahead, when we go into area study 2, we're going to learn about enzymes. The way they do their work, yes, you might think they need lots of ATP, so why haven't I got ATP in there? Enzymes actually don't. When you have the correct reactants, an enzyme will just join to them, attracted to them and join to them. So we don't have to put energy into this system. If anything, I guess the heat energy is enough to make the molecules move around, kinetic energy, but the main purpose of the heat is to separate the DNA ladder of our gene so we can create the new sides.